Okay, is this working? All right, thank you. Thanks, Dr. O'Hara. I think I've lived here just this month, 20 years in Houston, so I consider myself a Houstonian now, even though I grew up in Fort Worth. And it still surprises me, as long as I've been in Houston, I still haven't been everywhere in Houston, and so this is my first time to be on the HBU campus, and it's a pleasure to be here. Thank you very much for the invite. Dr. O'Hara asked me, since I am a little perhaps different from some of your other academic speakers, to talk about a little bit about how I arrived as a sort of an entrepreneur and company founder. Just real briefly, as was said, I finished biomedical engineering at Texas A&M in 1996. I guess when I started in 92, like many of you, I thought I might go to med school, and then I realized I was just too much of an engineer at heart, and I couldn't shake it. So got out after undergrad and worked for a couple years at different industry jobs, and one of those kind of led me down to JSC, so I lived in Clear Lake and working for NASA on a water quality instrument there. And then at that time I decided it was time, to go back to graduate school, and so I switched to kind of back towards my first love, which was chemistry, and wrote a lot of grants after graduate school, mainly through what's called the Small Business Innovation and Research Program, or SBIR program, which is a class of grants. 97% of the extramural funding that comes from NIH or NSF or what have you that funds your academic advisors goes to academia. The other 3% is mandated to go to small businesses. So we are all, in the small business world, we're clamoring for even a smaller pot of money to do research. But we, as you hopefully will see, we do a lot of interesting research. Base Pair is now not primarily funded by grants. It was in the beginning, but we have paying customers, which is a novel thing, and ranging from big, huge pharma companies that you've heard of, like Merck and Pfizer, all the way down to startups. And I'll explain the technology, and hopefully you'll see why it's a platform technology that's being increasingly used by lots of folks. But anyway, yeah, to round out the story, in 2012, because of the expiration of some other patents that were keeping me and my invention from, I couldn't practice my invention until a particular patent expired. But about the day after that expired, it's no coincidence that Base Pair was founded in 2012. And so now we're a company of about 20 people. We're down in Pearland, so effectively neighbors for Houston-wise. And yeah, we're having a lot of fun. As it goes with small companies, we're always raising money and seeking investors. But it's been a journey, and I have a 4-minute commute every day, so it was the first time I've driven into Houston in a while, and I don't miss it. So anyway, I'll get started. Just the outline of this presentation really quickly. We're going to kind of go out of order. The first 3 slides are little, what I call vignettes of ways we think we're making a difference. You study something for 15 or 20 years, you want it to actually matter and help people. And so we have 3 examples. Making a difference is really important to us as a company. And so I have 3 examples of how we're doing that amongst many, hopefully. And then I'll step back and explain what the heck an aptamer is. I don't think the laser pointer's working, so if something's not clear, just raise your hand or something. So I'll talk about what's an aptamer, how they're made, which is aptamer selection. What are the advantages and disadvantages of aptamers over antibodies? So that's the established competing technology. I'm sure everyone here knows what an antibody is. This, as I explained to Dr. O'Hara, this is a converted pitch deck that we often make to potential customers that need to understand the basis of the technology. So there are a few slides. I apologize. I've tried to remove most of the commercial stuff, but there's some advantages of the base pair against our competitors, and then I'll finish with a whole bunch of application examples, and this is kind of, I don't know, an engineering heavy talk, I'm an engineer, so I can't help it, and so it comes from my perspective. But if there's anything that's not clear, we can talk afterwards, or feel free to raise your hand. So our first little vignette, I guess everyone ought to be aware of the opioid crisis that we have going on in the country today. It's a very sad thing. I think last year, for the first time, more people died of drug overdoses, and most of those opioid-related, than in car wrecks in the U.S. But that, as sad as that is, the truly most innocent victims are newborns, and so kind of amazingly, 1 to 2% of babies these days are born addicted to opioids, and they. The mother typically doesn't want to admit that she's been using drugs or doesn't even know she has a problem. The opioid addiction problem is kind of creeps up on you, and as some of you may have heard in the news, just five doses and some people become addicted. But anyway, if you think about, I think it's 400,000 babies a year are born in the U.S., 1 to 2%, that's still too many. And what happens is they undergo withdrawal symptoms right about the time that the mother's discharged. So the mother may or may not know she's had a problem, the hospital may or may not have detected a problem, and then they send the baby home, and the baby becomes, in the worst cases, not just cranky, but shaking, convulsing. You may have a mother that's not in her best frame of mind, and it just makes for a bad situation. So we've had significant grant funding. We have another $1.4 million on the way to continue the assays that we've developed to detect opioid compounds and their primary urinary metabolites in babies' urine. So that's an exciting project, and there's a little more detail down the road in this presentation. We just published a paper in Biosensors and Bioelectronics on detecting nerve agent metabolites. So you may have, I don't know if a lot of you are pretty young, what was it, 2000? There was a sarin gas release in a Japanese train. So these nerve agents are used as biowarfare agents, and terrorists get a hold of them. In most cases, I mean, they're very deadly, and you foam at the mouth, and it's obvious you've been exposed. But low-level exposure for our first responders and military personnel have to handle these materials. They're often exposed. Well, the government and the CDC are worried about low-level exposure. So that's about all I can say about that, but I'm limited to what I can say about that, I guess is what I'm saying. But we've developed assays to detect these very small molecules. And then the main point of this slide is that the molecules themselves, you can tell VX and GB only differ by a single carbon atom, but these are the primary metabolites of sarin gas and GB acid. These molecules would either be highly toxic if you tried to inject them in a goat or a mouse or a rabbit, and it'd be difficult to elicit an immune response. So it's hard to get an antibody to small molecules like this. And then even if you did get an antibody, the ability to resolve them, most antibodies to compounds this small are cross-reactive. And then finally, we're working with a fellow down at the University of Central Florida named Bradley Willenberg, and he came up. Everyone, sure, we want to detect Zika virus and chikungunya virus in people, but he'd like to detect it before he'd like to detect the prevalence of the virus out in the environment, and so we're helping him make detection affinity reagents so that you can actually detect whether a mosquito out in the field already has Zika. And so the idea is he has a little feeder, with sugar water out in the field, and if when the mosquito drinks, a little bit of their saliva actually comes out, and if we make up the feeding trough and this little membrane that wicks up the sugar water, if we design all this properly, then you can count how many times a mosquito has fed and actually carries the disease. And then an extension of that idea is, which has gotten him a fair amount of money from the Gates Foundation, is that you can look at the abdomen of the mosquito, and if you have the right gold nanoparticles conjugated to the right aptamers, which is what we make, the abdomen of the mosquito can change from pink to blue and tell you whether it has Zika, whether it's carrying Zika virus or not. And so you can, every year when the mosquitoes start, spawning or whatever you call it, I'm not a biologist, but whenever they start multiplying, you can count the prevalence of mosquitoes that change color. So those are my little 3 vignettes, just to hopefully put technology shouldn't come before need, and so we don't like to just talk about technology in a vacuum. We actually want to do something with it. But here we go. So here's the technical kickoff. What is an aptamer? So aptamers are small, single-stranded bits of DNA or RNA, and they're typically only 20 to 100 bases. And because they're single-stranded, they're not this pretty double helix, right? They fold in 3D space. They look like spaghetti in 3D. And they bind their targets the same way that all biomolecules bind. They interact through van der Waals forces, hydrogen bonding, salt bridges, et cetera. But literally, aptamer just means a part that fits. So one of the inventors coined that term. And I don't know why I'm waving my hand when the laser pointer doesn't work. But the idea is you can make a synthetic library of very many different sequences. So if you take 40 nucleic acids and each position is A, C, T, or G, then the math is you have 4 to the 40th potential sequences, which is like 10 to the 23rd. It's close to Avogadro's number. Our default libraries are 32 bases long, which is 10 to the 19 possible sequences. So from a practical standpoint, you can't fit that many nucleic acids in a reasonable volume. So we start with a library of about 10 to the 15th different nucleic acid sequences, which is 1 million billion. So it's a big library, and I'll show you what we're going to do with it. So go back. So these are what are called 2D folding diagrams of nucleic acids. So if you've taken a class on RNA or something, they probably showed you a lot of these. There are different computer algorithms that predict the likely free energy of folding. And so when you minimize the delta G of folding, this is what you can predict a particular aptamer sequence to look like. But that's obviously flat on paper. That's in 2D. What they really look like is down on the bottom here. Up at the top is the protein thrombin, and you can see this tightly binding aptamer that was selected to bind to thrombin sort of fits like a puzzle piece in 3D space. It fits that bottom loop on thrombin, and that's actually a rather famous aptamer that has like nanomolar binding to the target protein. This is one of those salesy slides. What can aptamers be used for? All the slides will be made available, so you don't have to write down anything furiously. But basically, in the big sense, there's research use only. There are diagnostics and formal clinical diagnostics. There's veterinary diagnostics, and then there's therapeutics, and then there's overlaps in all those places, right? When people say all the time it takes $1 billion in 10 years to make a new drug, part of the reason is that all along the way, the drug companies have to develop new assays to measure the response of your body to the candidate drug, right? So there's a big business, not only in making new assays for research, but in case you didn't realize, there's a huge business in making new assays for drug development. So how are aptamers made? I mentioned, and I'll just walk, since the laser pointer's not working, I'll walk it through. So in step one, we have this soup, and that's in that triangle up there. That's the million billion, that's the 10 to the 15th different nucleic acid sequences. And then in the classic approach, that weird shaped gray blob is immobilized, say it's a protein, an important protein like VEGF. It's immobilized on an affinity column. You apply the library, and especially in the first round, and you'll see what I mean by rounds in a second. In the first round, the vast majority of things are not going to stick, right? They're going to not have the right shape to have any affinity for the target. So you throw away all the non-binders, but then you wash and you collect and you elute the binders, so that rare fraction of things that do bind. Then hopefully everyone knows what the polymerase chain reaction is. It's a way of amplifying the DNA that you did get back, and you reapply that to the library. You amplify it and then you reapply it to the original column having the target. So it's through these iterative rounds of selection, and then you increase the stringency. So you wash more, perhaps. You wash for a longer time. You might increase the salt. You can do things to make it such that you only get back the tightest binding species, and then you can monitor that process by various means, but at the end you sequence it. So when I started this out 12 years ago, we would have to clone the library members into E. coli and have them sequenced one by Sanger sequencing. So it would cost us about $1,200 to get 96 sequences back. Now we spend about the same amount of money and we get 1 million sequences back because of the what's so-called next generation sequencing. So none of you, except for, I would say, very specialized reasons, I doubt many of you will ever clone and sequence things to get something like this back. But the point being is that we go from 1 million billion and narrow that down to only about 1 million. So we go from 1 million billion down to 1 million, so we've dropped down the library diversity by 1 billion-fold, or 10 to the 9th. We then sequence that, and we do some specialized bioinformatics, and then we do some functional screening of the individual aptamer sequences. And so to the customer, what they want, they don't want to screen thousands of candidates to bind their target or to build an assay. They'd like us to get it down to 2 to 20 best aptamers. So you can think of that as the analogous thing. If you say, okay, I have an antigen in my research model in Drosophila, and it's a new antigen. There's no antibodies out there. I have to go to an antibody company. You don't want them to send you 1,000 candidate antibodies. You want them to send you just a couple, right? Or they send you a polyclonal and you don't know what's in there. The advantages of all this, so again, why? An aptamer is about 1/10 of the size of a typical antibody. A typical IgG is going to be 150,000 Daltons. An aptamer, once we're through with it, might only be 12,000 Daltons. It's completely synthetic, so it's just a sequence of 32 letters. So you can order it from IDT or wherever you order your PCR primers from. And you actually order it by email. You just email them 32 letters and say, I want this many micrograms back, and it might only be $70 or something. So it's infinitely renewable, and so with polyclonal antibodies, say you have a goat or a rabbit or a mouse as a source of polyclonal antibodies. Once that animal dies, your supply for that reagent is gone. And even monoclonal antibodies can vary from lot to lot in their performance. Because they're small, They can penetrate sterically into places that, that antibodies cannot. And maybe perhaps more importantly, they're made by in vitro selection. So again, kind of like in the sarin gas example, some molecules are so small that they don't elicit an immune response on by themselves. So you squirt a molecule into the bunny and it just doesn't, there's no titer for the antibodies. So even 6 months later, they're not making antibodies to the thing. People play tricks with small molecules. They conjugate them to carrier proteins like BSA or KLH, but often then the antibody only binds that conjugate. It doesn't bind the free small molecule. And because aptamer selection is an in vitro process, we can select aptamers in the end-use application. So we can select them in urine. We've done aptamers in horse urine for drugs of abuse in horses. Drugs of abuse like performance-enhancing drugs in horses. We can select aptamers in saliva for, say, like a THC roadside test. We've selected aptamers, well, you'll see all the examples, but the point is we can select them in non-blood matrices. They're also more stable than proteins. So if you want to develop a point-of-care assay in the developing world out in the heat, most of your antibodies on a lateral flow strip will denature. That means misfold. Aptamers are nucleic acids. They're going to attain that most favorable shape right as soon as you rehydrate them. So you'll see examples of where we've tried to develop, we've developed lateral flow tests using aptamers. And I kind of mentioned this before, that they're made by chemical synthesis, so there's no lot-to-lot variability and basically unlimited supply. This is another kind of salesy slide, but just real quickly to acknowledge, we're not the only company in the world that makes aptamers. There are five, six, seven, depending on, their focus. But we have 2 or 3 really main competitors that do what we do, which is custom aptamer discovery. We differentiate ourselves from them in a number of ways, but the main way, though, is I'll show you. We have a patented process on multiplexing this aptamer selection technology. So in the example with the 4 steps, that's the iterative selects process. You go through that, and it may take 3 or 4 weeks to get an aptamer to a single target. As a chemical engineer, I was kind of frustrated with the low throughput of that. And so now we can do, to cut to the chase, we can do 30 targets at a time. And so that's what we call multiplex selects. Basically, instead of just one gray blob on the column, you can think of having 30 different proteins on the column immobilized. So instead of throwing spaghetti at the wall and seeing what member of your library, what string of spaghetti sticks to that one protein, you can see what sticks to all 30. And the trick, of course, is how do you figure out which aptamer is going to which target? And that's the patent, which is basically a DNA barcoding strategy. In the very last step, we split out all the targets, and then we can deconvolute which aptamer is going to which target. If you're following through that, then we can take it a step further, and we add a little bioinformatics step on top of that. So the numbers in this Venn diagram, under protein 1, it's tough without a laser pointer, but under protein 1 it says 6278. So there was roughly 6,300 aptamer candidates that we think bind just to protein 1 and not to 2, 3, protein 2, protein 3, or protein 4, or the serum background, which is that blue lobe. And let me start over. You're probably reading on the left, but this was a real client, a pharma that you've heard of. And you can think of these proteins as loosely related. So say they're cytokines. They said they want aptamers to all 4 cytokines. But if you inspect the primary amino acid sequence of the 4 cytokines, you wouldn't expect them to have any shared epitope, at least not on a linear sequence base. But what that top blue arrow is pointing to is there were 19 aptamers that putatively bound all 4 proteins and nothing else in serum. So what that was telling us is that there must be sort of, even though you wouldn't guess it based on the amino acid sequences, there must be some sort of conformational epitope or shaped-based epitope that all 4 aptamers can bind. And that turns out to be really useful for pharma, right? So you can imagine, if you know how a sandwich ELISA works, you could have a pan binder, and we tested these, and 12 of the 19 did bind all 4 proteins. So you can imagine having on the bottom of your ELISA plate a pan binder that grabbed all 4. So you grab all 4 in this class of cytokines, and then you have independent detection reagents. How much of protein 1 was there? How much of protein 2 was there? So if you're familiar with pharma development, you can imagine a lot of reasons why you might want to do that. So I want to understand how much of a total inflammatory response was there, but then I want to figure out how much of it is attributed to which cytokine. As I mentioned, making antibodies to small molecules can be really challenging. And I sort of hinted at a lot of those reasons. Also, a lot of small molecules, if you can only do chemistry for mobilization at certain types of functional groups like primary amines, carboxyls, sulfhydryls. If the small molecule doesn't have any of those, then it's very difficult to immobilize. So you can't even conjugate it to a protein to boost your mouse or your rabbit. Or if you use a derivative of that small molecule, you may be taking away a seminal feature of that molecule. So it's no longer Prozac or whatever, aspirin or whatever you're wanting to make an aptamer to, or I'm sorry, an antibody to. Because, well, we can flip the whole process on its head, and rather than immobilize the small molecule to something or to a protein, we can immobilize the library and then just offer the small molecule. And we can loosely hybridize our library members, and only the library members that respond to that free small molecule come off the column, and that's what we collect. So we can play tricks in order to make affinity reagents in vitro without having to go through the limitations of any animal process. Another salesy slide. You'll see this in all the examples, but the take home of this slide is that we have a lot of assay development experience. So we don't just go through selects and then throw the candidates over the wall at the customer. We actually have a lot of assay development experience in-house. We have folks that worked at LifeTech or Invitrogen, if you're familiar with those companies, molecular probes. Our lab manager developed over 50 fluorescence-based assays at Invitrogen over the last 10 years. So we don't just, we like to do a lot of assay development with the actual aptamers. And so now we're getting into some specific application examples. This one is not a full assay for MRSA, but MRSA stands for methicillin-resistant Staph aureus, if you haven't heard of it. If you go to a hospital, you see signs all over the walls that say avoid MRSA, wash your hands. But MRSA is just one of many emerging bacteria that have emerging, what am I trying to say, antibiotic resistance. People sometimes, you can't convince them unless they see it, and seeing is believing. In this example, in A, it's kind of our negative control, but in B, we've spotted the MRSA protein. So the first step is to flow a biotinylated aptamer. If you don't know what biotin is, biotin's a small molecule. It's a vitamin. It's a covitamin that we all have, but it binds streptavidin, which is a protein, with femtomolar affinity. So it's almost a covalent interaction. So The biotin-streptavidin interaction is frequently used in lots of diagnostics, including ones you've had at the doctor's office. It's one of the nuts and bolts kind of things that routine clinical diagnostics are built on. So the aptamer at the end has this tiny little biotin, and it flows up the stream. We dip it in there, and then because we selected it to have high affinity for MRSA, it flows right past a negative control spot, which is just BSA, and then it binds to the MRSA protein. And the next step, the way you develop that pink color, is you dip it in streptavidin gold. So there's streptavidin stuck on gold. And this is not a pretty pregnancy test that you would buy at Walgreens, right? See the ugly line, but this was 4 or 5 years ago. I'll show you some prettier lines in a minute. And this is an example. So why is this important? We had a customer come to us that wanted to detect a molecule called dicamba. So dicamba, if you go in your mom's garage, she's got Roundup in the cabinet if she gardens. Dicamba is like the next generation Roundup. It's just a small molecule. It's a herbicide, but it's perhaps one of the most important herbicides in the world. And it's mainly used on soybeans that have been genetically engineered to be resistant to herbicides. So, you can spray your whole crop of soybeans and kill all the weeds, but you don't kill the soybeans. Say what you will about GMOs, it's really important technology for feeding the world, and it's increased our ability to feed people. This year, it varies year by year depending on global market needs, but this year soybeans will be the largest crop in the US at roughly 91 million acres. It's the 4th largest crop in the world. A lot of soybeans don't go directly to feeding people, they go to feeding livestock. But about 60 million, 2/3 of the US soybeans are genetically engineered to resist dicamba. But the problem is dicamba, it floats around. It's highly volatile. So, their answer, Monsanto's answer, or whoever, BASF's answer is to watch the weather and only spray when it's not windy. Well, and they tell you to download an app. That's not really satisfying. And actually, there's kind of, since this is HBU and I'm a Christian, I'll tell the story. There's kind of a Cain and Abel story in Kentucky last year where one brother damaged the other brother's crop and they killed him. The brother got so mad he killed his brother. So people have furious lawsuits about this. And you can imagine if you had soybeans next to $1 million vineyard and your dicamba actually drifted over and took out a vineyard, serious lawsuits have ensued. And so the estimated loss is about, well, you can see 1.1 million acres were damaged last year as a result of drift, and it's about $100 an acre. So the estimated loss is about $100 million a year just in dicamba drift damage to soybeans. So what we wanted to do is make a test. Part of the problem also is farmers don't just grow one crop usually, unless they're just a huge operation. They may grow corn and all this other stuff. So they fill their tank, they have spray tanks and big booms, different equipment, and they fill it up with the water and it has the pesticide in it. But now they want to they got to spray on some other crop, and if it's got any residual dicamba in the spray tank equipment, it could damage another crop. So it's very limiting. It's an important technology, but it's become problematic, and so we want to make a lateral flow test, which is, like a pregnancy test that a farmer can read, very simple readout, maybe a cell phone app to take a picture, and so to test for dicamba. And if you're not familiar with the way a lateral flow test works, we might as well go through it. And so the first lateral flow test was made against human chorionic gonadotropin. So if you're ever interested, it was an important step, right, to be able to know when you're pregnant. And that began in the 60s and 70s. And that's why today they're extremely sophisticated little bitty devices that you can buy for less than $10 at Walgreens. But you have a sample pad, and that's where, in the pregnancy test case, you would urinate. In this case, we're just dipping it in water. There's a conjugate pad, which has an aptamer on a gold nanoparticle. And then there's two test lines. There's a test line and a control line. At the test line, we have BSA, which is bovine serum albumin, conjugated to dicamba. So dicamba is that blue star. And then the gold particle with the aptamers, that octopus-looking thing, binds the test line. Okay? So if there's no dicamba in the sample, this is what's called a lights-off test, so it's a little counterintuitive. If there's no dicamba in your water, say you're testing your spray water, then you'll get a positive signal at the test line. If there is free dicamba, shown over here, then by competitive binding, most of the gold nanoparticle reagent gets occupied, and it flows right past the test line. So this is what the result looks like. So again, well, I call it a drug because I was sloppy in cutting and pasting. We've done this for other molecules. So on the left, there's no dicamba, so you get two lines. On the right, there is dicamba, you get just one line. You don't get the control line. Novel lateral flow tests for small molecules enabled by aptamer technology. That's what that is. And so we've also, as I mentioned, worked in weird sample matrices. On the left, we've worked with Stanford University to make aptamers to THC, actually the psychoactive form. As different states legalize marijuana, if you want to determine if someone's inebriated and too high to drive, you want to detect the psychoactive version of the compound. Unfortunately, if you use THC regularly, there's the carboxyl derivative, which is and it differs only slightly from this psychoactive compound. So from an antibody or aptamer standpoint, it's very difficult to discriminate between THC and the carboxyl long-lived metabolite that's in your body. Now, if you smoked marijuana 2 or 3 days ago, you're going to have that long-life metabolite in your saliva, but it wouldn't be fair for the police to say you're inebriated. So, it's different than with alcohol. If it's either still there or it's not. So we've developed aptamers to be selective between the active compound and discriminate from the long half-life metabolite. But more importantly, we did the selection in saliva. So because it's important, the same aptamers probably wouldn't work in blood, right? The environment that aptamers bind in and antibodies is important. When you buy an antibody from a catalog, they say it will work best in this buffer. We've also done work for the CDC on pork tapeworm. So these samples were actually literally fecal preparations, put a tapeworm in a blender, and we worked with those things. So because aptamer selection, again, is an in vitro process, we can work with gross bodily fluids, which is yay. So I'm kind of a technology buff, so this is another technology we use. We use different tools. We try to use the right tool for the job, right? We don't try to hit nails with a screwdriver or vice versa. This one is called microscale thermophoresis. There's a link there. Again, you can have the slides. But the idea is there's 2 lasers involved. There's one is an infrared laser that heats up the inside of a capillary and creates a thermal gradient in the radial direction of the capillary. Now, then you label, in our case, the aptamer with a fluorescent molecule, and you interrogate that with a different laser that excites the fluorescent dye. Just like every technology that measures binding, you want to somehow discriminate how much is if you've had biochemistry 101, like Michaelis-Menten, or how much of the bound complex is there relative to the two free species, right? And then you can work up a binding isotherm and say what the equilibrium dissociation constant is. So this is just a way, a fancy way of going about figuring out if it's bound in this thermal gradient, and then you turn the thermal gradient off and let things cool again, the bound things move back to the center of the capillary more slowly than the free. Okay. So it's just, it's a better way than calorimetry to do binding studies. And this is kind of what the data look like. You have a fixed aptamer concentration, and then you offer 16 different target concentrations, and you get a nice sigmoidal curve, which should mean something to you if you've had the right coursework. But the KD is sort of the half bound, half unbound. You can think of it that way. And so this is an aptamer that we made to noroxycodone, which is a primary urinary metabolite to oxycodone. So this is in urine. This is back for the babies. And we show that our aptamer binds with a nice binding constant of 43 nanomolar, which generally means you can develop an assay at least as good as the KD. You should be able to, and oftentimes about 10 times better. So we can detect very low amounts of noroxycodone, indicating that the mother's been using oxycodone. This is a table basically showing specificity. We build in selectivity into our process, right? So we can offer the off-target molecules to the library first, throw away anything in the library that interacts with that, and then grab back the aptamers that interact with the intended target. So the ones highlighted in green, those are the affinities showing the best specificity for the molecule. So for each of our aptamer candidates, in the first column there, you can see we've got a good aptamer to oxycodone at 15 nanomolar, but it also binds the metabolite. So that might be confusing. Rather, we can detect the parent drug only with that CFA 0072, but we don't detect the others. So it's a specificity table showing that we can make aptamers to our intended targets. Switching gears again, and I have several follow-on slides to this. Kind of a technology buff. I like different tools for different jobs. This is electrochemical sensing with aptamers. So if sorry, I don't have the laser pointer again, but if you look at that little gold surface and you imagine that spaghetti-looking thing is the aptamer, and at the distal end, that blue ball is a reagent called methylene blue. So it's a small molecule redox reporter. We can select aptamers to, say, the green ball, which is the small molecule target, say it's oxycodone, or any antibiotic you want to detect in blood, say. When the aptamer binds the target, the aptamers almost always undergo a conformational change upon binding the target, because they're loosey-goosey wiggling around in space, and then we've selected them to hug the target, and then they stay longer in that conformation. And so once he hugs the target, which is the green ball, the methylene blue is closer to the electrode surface, and we can sense that as a current change in our electrochemical device. And the promise of this is that the devices that do the readout, since there's no optics, you don't have fluorescence and microscope objectives and laser diodes and all this stuff, you can do inexpensive mass production, right? There's thousands of semiconductors in here that can make these same kinds of measurements. And also because it's not optical, you can read it in whole blood or other highly scattering samples. Think of pap smears, saliva. Biology is not optically transparent by nature, right? And so there's also the potential of reversible sensing in long-term implantables. So, and these are real data actually from Kevin Plaxco's group out in California. But you can see that you can track real-time concentration swings as the drug is dosed into a lab animal. One caveat to all of this is eventually these types of sensors get walled off from the body. So somebody has to solve the sort of long-term biocompatibility problem to have, full long-term implantables. But for cases of, say, when you're in the ICU for just 3 days, an implantable sensor like this could be, life-saving. So here's an engineering slide I won't bore you too much with, but most semiconductor transistors are kind of made this way nowadays. There's what's called a drain, a gate, and a source. And I'll just say that the gate is generally where they put the biomolecular recognition element, which in my case is an aptamer. You could put an antibody there, but I'm against antibodies apparently. Not really, and that's not true. Where they do a great job, they should keep doing a great job. And but that's the engineering part of it. This is the chemistry part of the same paper, and this is using our aptamer. We have an aptamer to tenofovir, which is the frontline HIV drug in the world, and it works really well in a variety of platforms. But basically, with this sensor, they show about 3.5 nanomolar limit of detection, and it doesn't bind other related small molecules. These are other antivirals, et cetera. So the aptamer is selective as well. Same kind of story, just a little bit different sensor architecture. This was in Nature Scientific Reports. They used our tenofovir aptamer, and they get very similar limit of detection of 1.2 nanomolar and a linear range of 1 to 100 nanomolar. So, and one of the main points in the paper was to optimize sort of the steric spacing between the aptamers. And then these are some more of our friends, different friends at the University of Central Florida, who have a special, like extra add-on organic layer to their sensor they call a PDOT, which is just a particular polymer that, and I don't fully understand it, but it extends the limit of detection down to 90 picomolar and broadens the linear dynamic range, over 5 logs. So 90 picomolar up to 900 nanomolar. So 5 logs? Yeah. 3 logs anyway, 4. And you'll have these slides. They have the references. You can read the papers if you're interested. Did someone raise a hand, or were you just stretching? Okay, okay. People want to hear about flow cytometry. Has anyone ever used flow here? Okay, sure. Okay, well, and it doesn't really matter. So, flow cytometry is a way to rapidly count a large population of cells, like millions of cells. And if you end up, working in the med center over at MD Anderson or something like that, you'll end up using it. And so typical, the more conventional way is to have a fluorescently labeled antibody. You stain your cells, then you run it through the flow cytometer, okay? So instead of using an antibody, these folks developed an aptamer to a new surface target, and then up top, they demonstrated that, yes, you can, so what you're looking for is these separation of populations of cells. So there's 2 hot spots kind of on the top right? There's cells that there's a lot of surface antigen being detected, and then there's a lot of cells that aren't being detected. But, you're looking for clusters. That's great. So basically, they just proved that you can make an aptamer to a target that already has plenty of good antibodies for it. So it's more of a gee whiz, but what's below, though, is much more powerful. So imagine doing whole cell selects, so instead of that protein, that gray blob on the affinity column, instead of that as your immobilized target, a single pure protein, say I don't even know what if this population of cells, I don't know what the marker is on it. I just know that its phenotype is cancer. All right, so I have a cell culture or cell line, and its phenotype is healthy, and I do healthy in air quotes because, what does healthy mean? But we do know that this cell line has a phenotype of cancer, or it's been infected with HIV or HCV. It doesn't matter. So without any a priori knowledge of what the biomarker might be, you can bathe this group of cells in your library and come up with an aptamer. Now, what's it binding? I don't know, but my assumption is that some surface markers on this cell population have got to be different because they have cancer and they communicate with the rest of the body differently. So that's what we call whole cell or naive selects. And what this paper shows, and there's a lot of examples of this, is you go through that process, and then they call it JHIT2. It's the orange population. This is a different way of looking at those scatter plots. But you see how that orange distribution is shifted to the right? It means that aptamer detects something on the cells that's different from the control cells. See, all the control cells reacted to the labeled aptamer. So maybe I wasn't clear. Now you got JHIT 1, 2, 3, 4, 5, 6, 7. They're all labeled with a fluorophore. And you bathe, this is after the select process, you've got these candidate aptamers. They're clones. They're just specific sequences of 32 bases with a fluorophore on it. You bathe the cells, the normal cells are on the right. You bathe them in any aptamer, and there's no shift. But if you bathe, after you've gone through the selects process, you bathe the aptamers in you bathe the cells in any one of your 7 aptamers, and you get a shift. And especially JHIT2 is detecting something that's very different about the cancer cell than the normal cell. So what does it bind? Well, now you can go figure it out. You can put your aptamer on magnetic beads. You can do a membrane prep of all the proteins, pull down that protein and send it off to your mass spec core lab, which MD Anderson probably has 3. And 2 days later, they tell you what the protein was. And they say, oh, this novel protein you've discovered is indicative of this phenotype. And so you've gone from a hypothesis and a phenotype and a guess to novel biomarker discovery. Does that make sense? Just real quick, we're getting to the end. This is showing that aptamers can also be used in immunohistochemistry or fluorescent staining of tissues. So this particular target is called LO37. It's a peptide that's 37 amino acids long, go figure. LO37 is actually really interesting, though. It's part of your ancient immune system. Before we had an adaptive immune system, we had just these like cationic peptides floating around in us, and I don't know what we were at that time. I'm just this is old. This is very ancient. And the cationic peptides, their main purpose is to sequester foreign viral DNA or RNA because they're cationic. They have lots of lysines. And so if there's small snippets of viral DNA or loose genomic DNA or bacterial DNA floating around, it was meant to sequester that. Well, I have a good friend. He's chair of the Department of Dermatology in Switzerland, in Lausanne. he used to work at MD Anderson. He lived around the corner, and our daughters were having a birthday party together, and then we figured out we had a lot of common interests. And so now we've been working on this off and on for like 10 years. But Michelle and I have done several studies on making aptamers to LL37. We made aptamers to the LL37. Why are we doing this? Michelle figured out that even though it's part of your ancient immune system, LL37 is still hanging around, and now it's implicated in autoimmune diseases. And his theory is, and others, actually psoriasis is more of an autoimmune disease than anything. So what happens is LL37 sequesters your self-DNA. When cells lyse, it sequesters your genomic DNA, and then it starts these conglomerates of LL37 and your own genomic DNA start turning into granules, and it looks like local inflammation. And so we've measured the IFN gamma as his primary readout, or IFN alpha, sorry. And with our aptamer, we can stop that process. So we dose, and it's a local delivery, because if you have psoriasis, you know that the skin is locally damaged. And so we think we can deliver the aptamer as a therapeutic locally. It goes in selectively into the damaged skin, so you've got a local delivery mechanism there, and then it binds LL37 before the genomic DNA can, in the next round of cell lysis, can be bound. And we actually think we have a lead drug candidate for psoriasis. So if you have money, we're trying to raise money around that. This is pretty complicated, but all I'll say is I had a grant from the National Cancer Institute to make aptamers specific for phosphorylated peptides. So again, the theme is because this is aptamer selection is an in vitro process. We can ERK1, where it says aptamer, ERK1 and ERK3, it should say parent protein. Those ERK1 and ERK2 are common cancer biomarker proteins. That peptide, that's about 20 amino acids, is a daughter peptide. It's contained within the whole ERK1 sequence. The underlying big T is phosphorylation at the threonine. We can make aptamers that bind that peptide, but not the one on the right that doesn't have the phosphorylation. Similarly, we can discriminate between threonine phosphorylation versus the phosphotyrosine, and both of those exist in nature. So what would anyone use that for? Mass spectrometry is often used to quantify such biomarkers. So you take a cell line, lyse it, and then we put our aptamers on magnetic beads. The aptamers bind the labeled peptide. Well, in this case, it wouldn't be labeled for mass spec. You don't need it for mass spec. But we show it visually by fluorescence. And so we show the loading of the sample. We show the supernatant, which is depleted, and then we elute the labeled peptide again, and then we quantify it by mass spec just to show the how do you say, proof of principle. But the R squared of that was almost 1, so pretty amazing assay linearity and sensitivity. Hard to get into all the details here, but I think I'll skip this. Aptamers have been used in drug delivery. This is not my work. This is just to make the point that if you're familiar with siRNA and the difficulty in delivering siRNA, 10, 15 years ago, siRNA was going to be the next big thing, drug class that saved us all. It turns out that it's very difficult to get siRNAs into cells. Other payloads have been conjugated to aptamers for drug delivery, mainly for cancer. So You can imagine conjugating an aptamer to a specific cancer biomarker to a common cancer drug like doxorubicin or something and selectively killing cells. Those are these sorts of papers. I currently have, if you haven't been following, the Nobel Prize in Medicine was recently won around cancer immunology. What's his name? Allison here at MD Anderson. I didn't mean to associate my work with his. I have a new grant for studying, making aptamers to canine immuno-oncology markers. It's increasingly understood that canines are a much better model for cancer studies than, say, mice. Dogs actually get cancer, right? And we have to manipulate mice quite a bit to actually even make them get cancer within their normal lifetime. But there's a dearth of reagents out there for a lot of the antibodies that you would use in a mouse study and then translated to a human study just don't exist for the canine versions of the proteins. So we're just using our factory to make a bunch of affinity reagents to important canine proteins just for the purpose of translational medicine and oncology studies. But separate from all that canine stuff, here's some nice papers on aptamers in cancer immunotherapy. More just kind of gee whiz stuff. You can conjugate an aptamer to fluorophores or quantum dots, and if you induce a tumor in a particular region of an animal, you can see that the labeled aptamers do accrue at the site of the lesion. We have a customer over in Italy who works in the area of dental implants, and he used one of our aptamers to fibronectin, which is an extracellular matrix protein, to recruit osteoblasts. So if you want to grow dental tissue in a tube, this would be like your foundation layer. And this is all really complicated. I think I've probably used all my time. I think to summarize, hopefully, I've made the case that aptamers are an alternative to conventional antibodies in pretty much any affinity reagent situation one might envision. We can make aptamers to small molecules and non-immunogenic compounds, toxic compounds. I've tried to give a number of strange examples all over the place, but examples where they work just as well as antibodies in various tools. And then that last part, some sales stuff, I'll spare you. So, that's it.